final homeostatic mechanism that we're going to look at today is glucose regulation. So before we start off, very basic review. Sugar is glucose, and we need a constant supply of that glucose in order for us to complete cellular respiration, in order for us to get energy. Now, normally we get glucose through eating, and the easiest way for us to get that through eating is by eating good carbohydrates. They're really readily broken down into glucose, digested, and absorbed into the blood. So naturally, after we eat, our blood glucose levels begin to increase, and after a long period of time without eating, our blood glucose levels, BGL, come back down. Okay, so when we're talking about where this glucose, if we eat way too much, goes, glucose can be stored in the form of glycogen, and glycogen is simply lots and lots of glucose stuck together. When we're talking about glycogen, 20% is stored in the liver, 80% is stored readily in muscle cells around the body, so that if you need that energy, you can readily access it. And this process is known as glycogenesis, where we create glycogen. All right. Now, in terms of the body, normal blood glucose levels range between 4 to 6 millimoles per litre. This won't appear in your exam though. Something that's worth noting, however, is the unit for blood glucose, millimoles per litre. So, very quick flow diagram, but if your blood glucose levels are high, your body will store it as glycogen, so lots of glucose stuck together. And then when your blood glucose levels get low, your body will break the glycogen down into glucose. So how do we do this? So again, glycogenesis is glucose into glycogen. Glycogenolysis is now where we take that glycogen, again, so I'll highlight the glycogen in both words here, and lysis means breakdown. So we're breaking down glycogen and we're forming it back into glucose. The final one for us is gluconeogenesis. Gluconeogenesis is where we take so fats or lipids. We can also take protein structures like amino acids, and we can turn them into glucose. Now, these three different forms of glucose and forming glucose and breaking down glycogen into glucose really help us with this blood glucose level balance. So, everything is controlled by the pancreas, and the pancreas here is really important in control of two really important hormones known as insulin and glucagon, which we will talk about now. So within the pancreas, we have both alpha and beta cells, and it is these alpha and beta cells which are responsible for the release of insulin and glucagon. We also, within the pancreas, these alpha and beta cells are located within groups of cells known as the islets of Langerhans. So let's have a look at insulin to start. So insulin is released by your body in response to a high blood glucose level. So think post-meal. Okay, so you've just eaten a really sugary meal. This is when we have our highest blood glucose levels. This is detected by chemoreceptors, so again, receptors that detect chemicals, and very specifically here, we're talking about the beta cells of the islets of Langerhans. These are found again within the endocrine organ that we just looked at, the pancreas. Now, these beta cells within the islets of Langerhans secrete insulin into the blood, and what they end up targeting is all of our body cells, the liver, and skeletal muscle. So three distinct effectors, each with a distinct response. So in terms of all body cells, if we have too much glucose running around in our blood, all our body cells are told to help out and take in glucose. In the case of the liver, we have the process of glycogenesis, where we are taking glucose, and we're converting it into that stored form, glycogen, and we're storing it in the liver. So about 20% is stored in the liver. We can also have a process known as lipogenesis, and this is where we take that glucose and we store it as adipose tissue in the liver itself. Finally, in some cells, we can also have protein synthesis, so the creation of protein with this used energy. Overall, all of these different responses are going to lead to an increased level of blood glucose. No, that's not quite right, is it? It is going to be a decreased blood glucose level, okay, decreased BGL in response to high blood glucose level. So this is an example of a negative feedback system. Contrary to insulin, we now have glucagon. Glucagon, again, is released thanks to a low blood glucose level. And before I forget, this is going to eventually lead us to a negative feedback system where we increase our blood glucose. So very similarly, detected by chemoreceptors within the islets of Langerhans, but this time the key part that I will highlight to differentiate is that they are detected by alpha cells. And it is these same alpha cells that secrete glucagon. So I'm just going to highlight all the differences in green on this slide. The effector remains much the same, so again, all body cells, the liver and skeletal muscles. And the effect this time is glycogenolysis in the liver and skeletal muscles. This is that breakdown of glycogen into glucose. It'll also lead to gluconeogenesis, so where we break down those fatty molecules and turn them into glucose. And there is a very mild effect where we promote protein breakdown, but we don't want to break down too much protein within the body because we still need these proteins to either help with chemical reactions, such in the form of enzymes, or we don't want to break down muscle as well around the body. What this results in overall is an increased blood glucose level as we break down our stored forms of glucose and we release them into the blood. So we've now got two other examples, I will say, being cortisol and adrenaline, which can influence blood glucose levels. So the main two that we've spoken about so far have been insulin and glucagon, which are regulated by the pancreas. But in the case of cortisol and in the case of adrenaline, they're no longer regulated by the pancreas, but they're regulated by other systems in the body. So let's start by looking at cortisol. Cortisol is released in response to a low blood glucose level, so over a long term generally. And this is a form of stress that is placed on the body because the body doesn't have enough energy. And so this is detected by the hypothalamus, and what it triggers is the adrenocorticotropic releasing factor. And this ACTH-RF is released into the hypophyseal portal system, targeting the anterior pituitary to release adrenocorticotropic hormone, ACTH. ACTH is not cortisol, but what it does is ACTH travels through the blood and it acts on the adrenal cortex. And when it does, the adrenal cortex is triggered to release cortisol into the blood. Cortisol, similar to insulin and similar to glucagon, is going to target all body cells, the liver and skeletal muscles, and it's going to lead to 4 distinct responses. First of all, glycogenolysis, same as in glucagon, where we end up releasing and breaking down glycogen into glucose at the site of the liver and skeletal muscle. We can increase the rate amino acids are moved to the liver. We can also increase the movement of lipids from fat to the liver, and we can have gluconeogenesis in the liver itself as a response of that increasing rate that amino acids and lipids are moved to the liver. What this will end up doing, again, our stimulus has been low blood glucose level. Cortisol is working to increase that blood glucose level to have a negative feedback system. As we talk about the adrenal cortex, okay, the role of the adrenal cortex, again, is to just help us to regulate those blood glucose levels by releasing cortisol, and cortisol is going to stimulate those 2 processes, being glycogenolysis and gluconeogenesis. Final feedback loop for today, and our final real example is adrenaline. Now, adrenaline, again, is released in response to low blood glucose levels. Again, it's detected by the hypothalamus, and this time, instead of being a immediately hormonal response, the hypothalamus sends a message along sympathetic nerves to the adrenal medulla. Now, the adrenal medulla is then going to allow for the release of adrenaline, and similar to our past 3 hormones, it will target all body cells, the liver and skeletal muscle. Glycogenolysis will then occur, and this is going to increase blood glucose levels. So again, glycogenolysis, breakdown of glycogen into glucose. So the very final thing that we will talk about today, I'll just skip through these feedback mechanisms, is the liver. Okay. Now, the liver has a very large vein. You can see it in blue down the bottom here. I'll just run my highlighter over it now. This is the hepatic portal vein. And what it does is it carries blood straight to the liver from the stomach and the intestines. And one of the main jobs of the liver is to remove glucose from the blood, as we've just spoken about. And it can also remove and store glucose as glycogen or lipids. Now, glucose can continue to circulate to other body cells because, again, it just passes through, but this liver is a really important site of storage for us. Again, about 20% of all glucose can be stored in the liver.